Hello everyone, welcome to Biolessons. In today's class, we are going to discuss about sliding filament theory, which is the most important topic from the chapter Movement and Locomotion. Have you ever wondered how we are able to walk, run, or even blink our eye? Every movement, be it tiny or big, it requires muscle contraction. And whenever we talk about muscle contraction, sliding filament theory comes into the picture. Basically, sliding filament theory explains how muscles shorten during contraction. Okay? And it was given by Andrew Huxley, Rolf Niedergert, Hugh Huxley, and Gene Hansen. Basically, they were two teams who came up with the idea of sliding filament theory. Let us now have a quick recap about the structure of sarcomere and the contractile proteins, which is the prerequisite before going to the sliding filament theory. Now, sarcomere is also called as a functional unit of muscle contraction. It is the region of the myofibril that lies between the two Z lines. Okay, and the light and dark or the striated appearance of the myofibril is due to the specific arrangement of the contractile proteins, that is actin, which is a thin filament, and myosin, which is the thick filament. Okay, now this is your Z line, and these are the actin molecules which are attaching itself to the Z line, and this is the myosin molecule. Okay, so the light band refers to the I band or isotropic band, is made up of primarily actin. The dark band is made up of myosin plus actin overlapping on the ends. The middle light zone present in the dark band is called as the H zone, where there is no actin overlap on the myosin. Okay, and the dividing line, the middle dividing line of the H zone is called as the M line. So this is a quick recap about the sarcomere structure. Let's now come to the structure of actin. Now, actin protein is made up of three components: two molecules of F-actin, which are bound helically to each other, two molecules of tropomyosin, which runs parallel to F-actin and is present in the groove of F-actin, and troponin, which is a trimeric protein and acts as a regulatory unit. Okay, now you should know that this F-actin is a polymer which is made up of many monomeric unit of G-actin. Okay, so that's a review about actin structure. Now let's come to the myosin structure. Myosin molecule is made up of many myosin molecule, which is also referred as meromyosin. Okay, now it is made up of two heavy chains and four light chains. Okay, the heavy chains, they twist along each other and make up the tail part, and they are free to one end and they make up the globular head there. Okay, the light chains, they attach to the globular head region. Okay, now this tail portion is also called as light meromyosin, and the head part along with the short arm together called as a cross arm is also called as a heavy meromyosin. Now if you see, the globular heads, they possess the important regulatory sites, two regulatory sites, one for actin binding and another for ATP binding. Okay, this ATP binding is essential for the cross bridge formation in the muscle contraction. Okay, clear about this? Let's now go through the steps in muscle contraction. Okay, the brain sets up an intention to walk. What is it? It is the signal. So the brain sends the signal as a nerve impulse to where this nerve impulse is sent to the motor neurons. These motor neurons are nothing but the neurons that are associated with the muscle fiber. So this is the motor neuron, this is the muscle fiber, this is the plasma membrane of the muscle fiber, also known as sarcolemma. So this motor neuron and the muscle fiber, Together, they are known as the motor unit. And the gap between the motor neuron and the sarcolemma of the muscle fiber, that is known as neuromuscular junction or the motor end plate. So what happens after the neurons they receive the signal, they release a chemical messenger called as the acetylcholine. This acetylcholine, it binds to the receptors that are present on the surface of the sarcolemma, okay, or the plasma membrane of muscle fiber. So this acetylcholine released binds to the receptors and generates an action potential on the sarcolemma. Clear? Action potential which was generated causes the release of calcium 2 plus ions from the sarcoplasmic reticulum. What is sarcoplasmic reticulum? Endoplasmic reticulum of the muscle fibers is called as sarcoplasmic reticulum. Okay. So now this calcium 2 plus ions are released into the protoplasm of muscle fiber, also known as sarcoplasm. Here the calcium 2 plus ions, they play the magic. Okay, what do they do? These calcium 2 plus ions go and bind to a subunit of actin. Okay, which subunit does the calcium 2 plus ion bind? It binds to the troponin subunit. Okay, very important. Calcium 2 plus ions bind to the troponin subunit of actin molecule. Okay, now this binding of calcium 2 plus to the troponin molecule shifts the tropomyosin molecule a little bit. And what does it do? It unmasks the active site present in the actin molecule to bind with the myosin molecule. Okay, so now the active sites of the actin molecule are free or exposed and it is ready to bind with the myosin molecule. Okay, so this is the myosin molecule, this is its tail part, this is its short arm, this is the head region. We know that the head region of the myosin molecule, it possesses the two regulatory sites, one for binding ATP, one for binding the actin. So now this head region of myosin can bind with the actin filament. So this is called as the cross bridge formation. Let us now study the cross bridge formation in a little more detail. Let us now look at what happens in the cross bridge formation. Okay, this is your actin molecule, this is the myosin molecule, this is the tail part, short arm, head part. We know that the head part has two regulatory units, one for the actin binding, another for the ATP binding. Here the ATP molecule that is bound on the myosin head, that is hydrolyzed. This hydrolysis of ATP gives the energy for the binding of the myosin to the actin molecule. Okay, actin has already exposed its active sites, so the actin is bound to the myosin here. So this results in the formation of cross bridge. Okay, so what does this cross bridge do? This cross bridge utilizes the energy and helps in gliding of the actin molecule. So the actin molecule now glides a little bit or slides a little bit. So where does it slide? So the actin molecule are pulled towards the center of the A band. That is known as a power stroke. Okay, so the actin molecules are pulled. Along with that, the Z lines attached to the actins are also pulled inwards, and this in turn causes the shortening of the sarcomere or the contraction. Clear? After that, what happens? The ADP molecules and the phosphate molecules that were utilized to provide the energy are released out, and the myosin molecule now comes to the relaxed state. Okay, cross bridge is broken at that stage. Then what happens? Again a molecule of ATP binds to it and the cycle repeats itself. And until what time the cycle repeats? The process continues till the calcium 2 plus ions are pumped back to the sarcoplasmic cisternae. As long as all the calcium 2 plus ions are pushed back to the endoplasmic reticulum, the process continues in a cyclic manner. Okay, ATP is hydrolyzed, again the cross bridge is formed, and then again that ADP and phosphate are released, again a new ATP molecule is attached, and the process continues. Clear about it? So we looked into what happens in the cross bridge formation. Let us now see what are the changes that take place in the sarcomere during muscle contraction. This is the image of sarcomere in the relaxed state, Z line, I band, A band, this is the S zone, and the middle we have the M line. Okay. Now, because of the cross bridge formation, there is sliding of the actin over the myosin filaments, and the Z line is also pulled inwards. So look at the image here. You can see the Z line is shifting inwards. Okay. And what is happening to the edge zone? The edge zone is also decreasing in size. Okay. Now during the maximally contracted stage, what is happening? The edge zone is diminishing. But is there shortening of actin filaments? No. Both the actin filaments and the myosin filaments are not getting shortened. So what is getting shortened? As you can compare, the I band is getting shortened, the edge zone is getting shortened. Okay? Is that clear? Let us now recap everything. At rest condition, the actin molecule, its active sites are masked or covered. Why? Because troponin molecule is inactive. Okay. So tropomyosin blocks the myosin binding site. So actin and myosin cannot bind with each other. Cross bridge cannot form. Then what happens? Brain decides to work. So stimulus comes. Okay. This stimulus causes the release of calcium 2 plus ions from the sarcoplasmic reticulum. Calcium 2 plus ions goes and binds to the trimeric protein. What is it? Troponin. So it is activated. So this binding of calcium to the troponin molecule shifts the tropomyosin molecule slightly. What happens now? Active sites present in the actin are now exposed. So actin can bind with the myosin molecule. Cross bridge formation takes place. During the cross bridge formation, ATP is hydrolyzed, and this hydrolysis of ATP gives the energy to form the cross bridge, and this formation of the cross bridge causes the sliding of the actin molecules over the myosin molecule, which is also known as the power stroke. Okay, this sliding of the actin molecule over the myosin molecule causes the inward pulling of the actin molecules towards the edge zone, and this causes the shortening of the sarcomere. Are you clear about this? So this is the most important part of this lecture. So what actually changes during muscle contraction? By now you must know that during muscle contraction, there is just the sliding or the gliding of the actin filaments over the myosin filaments. There is no shortening of actin or myosin. Okay, both of them has intact length, right? So what changes? The length of the sarcomere decreases. Okay, the I band or the light band, their size decreases, and the H zone size also decreases. The H zone diminishes during the maximum contraction. Okay, and what happens to the A band, the dark band, or the anisotropic band? There is no change in their size. Okay, and the length of actin molecule and the myosin length also remains intact. It does not change. Very, very important. Okay, I hope you are now clear with this. So we covered the most important part of the movement and locomotion chapter today, that is the sliding filament theory. I hope that you enjoyed the session today. If you have any doubts regarding this, Please feel free to comment below. I would be happy to answer. Also, if you want to check out more on the structure of sarcomere, I am attaching here with the link for that video. You can go through it, okay? And let me know if you have any queries to that also. Thank you, everybody. Have a very good day.